Good morning, good afternoon, and welcome to today's seminar. We are happy to have Dr. Hela Rouhou from Tunisia, from the Regional Research Center on Horticulture and Organic Agriculture with us. Hela is in charge of the cucurbits breeding valorization programs. Hela has she did her graduate studies in Spain, in Zaragoza, and she also has a diploma in biotechnology and seed production from the National Agronomic Institute of Tunisia. As I said, she is in charge of genetic resources of cucurbits, melon, watermelon, pumpkins, and bottle gourds. But her main responsibility is breeding melons. That's the topic of her presentation. She also told me that she's, as of a few months ago, she's in charge of the onion breeding program too. So Hela was selected as a fellow of the African Plant Breeding Academy, and in 2024, she was also chosen as a fellow for the third cohort of the Arab Women Leaders in Agriculture. So, Hela, I appreciate so much that you volunteer for this seminar, and it's been nice to interact with you on the way to this seminar, and we look forward to your presentation, okay? I'm gonna stop sharing here and give you the floor.
Thank you, Carlos, for the introduction and for giving me the opportunity to present the results of my research. And thank you all for being here. I will share my screen.
All right, we are good. All right.
Okay. So hello everyone, and welcome to the webinar. As mentioned by Carlos, my research focuses on the conservation and valorization of local genetic resources, mainly cucurbit crops, melon, watermelon, buttergourd, and pumpkins. And today I'll talk specifically about my melon breeding that I'm managing. So I will summarize a bit of the journey with the success and achievement, but also the different challenges faced in my Tunisian context, starting from the germplasm selection to the hybrid development. So before diving into today's talk, I'd like to briefly present my institution. It's the Regional Research Center on Horticulture and Organic Agriculture. We are located in the center east of the country, in Sousse City, more precisely in Chat Mariam, along the Mediterranean coast. It was created in 2006, so next year we have our 20th anniversary. It is a small research center with 18 researchers in different disciplines, and the focus of our research is on horticultural crops, their production and protection for a sustainable agriculture. So the outline. So the outline will be as follows. I will give you an overview. I don't see the slide.
Yeah, maybe click on the screen before moving. We still see the slide of the center, Hela. Okay.
Yeah, it doesn't move. I don't know why.
I'm gonna stop sharing and reshare again.
Okay.
There you are.
Okay.
Yeah, the
outline would be as follows. I will give you an overview of the local context and why we are conducting this research on melon breeding, to show you also the great diversity found in our local land races. Thirdly, I will talk a bit about the microassisted selection that we are doing here for the major biotic stress, mainly for diseases and insects. And finally, with the heterosis and hybrid breeding, to show you the ongoing work of crossing and evaluating hybrids. So for the context, melon is a vegetable crop of high nutritional value. It's very rich in vitamins and antioxidants, such as polyphenols, flavonoids, and carotenoids. And Tunisia is one of the richest Mediterranean countries in terms of plant genetic resources, including land races and wild relatives, especially for the cucurbit crops and also for melon. So why we are working with land races and why local germplasm? Because they are well adapted to the local growing condition and offer a valuable source of genetic variability. So exploring their diversity will allow us to identify promising traits such as disease resistance or stress tolerance and fruit quality that I will give you the results later, so that can be leveraged for breeding efforts. So comprehensive evaluation of these genetic resources is essential, both for their conservation, because if it is not conserved, they are threatened by the genetic erosion. So we have to conserve them in a long-term conservation, but also for their valorization and their use for sustainable agriculture. So the main goal of doing the breeding program here in our research center is to develop resistant and highly yielding variety, starting from the great diversity found in our local germplasm. And as we can see here in this picture, the huge phenotypic variability that exists in the collected land cases. And this is the start point of our breeding program. So the main steps, we start firstly with the germplasm collection. So we collected several land races and several seeds from local farmers in different regions where we have melon growing in Tunisia. Then we started doing the genetic diversity using phenotyping and genotyping analysis in order to have a detailed agromorphological and molecular characterization. Then we started doing the screening for resistance to the major biotic stresses in order to identify the source of resistance to the main biotic stresses here in Tunisia. And then when we identify source of resistance and we evaluate the extent of the genetic diversity existing in the local material, we started making the crosses and the breeding program properly. So for the genetic diversity, we used phenotypic traits and also molecular markers. For the phenotypic traits, we used several agromorphological traits, quantitative and qualitative traits related to the flower, to the leaf, to the fruit and to the seeds, and also fruit quality was evaluated because it's the most important trait for the consumers, where we evaluated the total soluble solids measured by the degree Brix, and it measured the sweetness of the fruit and also firmness, which is an important criterion for the consumer and also for the farmers because it influences later on the shelf life of the melon. Also, molecular markers of SSR microsatellites type were used for our study of the genetic diversity. So for the phenotypic analysis using the phenotypic traits, we were able to cluster the whole collection in two different groups, two clusters. We have the big cluster, the big one, which is composed of the sweet melons, and the small cluster, which is composed of the non-sweet melons. So what is the difference between both clusters? So we have firstly different botanical groups that this analysis allowed to identify 5 different botanical groups, which are Inodorus, Reticulatus, Cantaloupensis, Clepsus, and Chatti. So what is the difference between each botanical group? Because each botanical group has its characteristics, and we can distinguish between each botanical group by the specific characteristics for each botanical group. So here for the Inodorus, for example, which is the yellow fruit here, it is not netted, it's not, the skin is not netted, it is non-climacteric fruit. It is moderately sweet and is very appreciated here in Tunisia. We have the Reticulatus botanical group, which is characterized by the netted skin. It is very sweet. It has an aroma when it is ripped. The fruit it smells a lot in the field, and it is andromonoecious. And we have the Cantaloupensis botanical group, which is the cantaloupe variety, which is the most famous here in Tunisia and also in the Mediterranean basin. It is characterized by the ripped in the skin. It is globular. The weight, it does not exceed 500 grams, and it can be netted or not. For the non-sweet melons, which are Flexius and Chatti group botanical groups, the Flexius is also called the serpent or snack melon. It is consumed here as a salad, and it is harvested when it is green in immature physiological stage. And we have the Chatti botanical groups, which is shorter than the Flexius one, and it is ovale, it is rough and sometimes with fine hair. So for using the molecular markers, the genotypic analysis allowed a more precise clustering. So here we have the different genotypic clusters formed using the different molecular markers, and we have distinct clusters genetically distant, and that will be used later once we're gonna start making the crosses. And both analyses using phenotypic or genotyping analysis showed the great diversity in this local germplasm and the extent of this variability. So we have major biotic stress that affects melon crop here in Tunisia. We have majorly the soil-borne disease, Fusarium wilt. We have the aerial-borne disease, powdery mildew, and the aphid, which is vector of several viral diseases in melon. And major genes of resistance exist for these biotic stresses. We have, for example, FOM1 and FOM2 genes, which confers resistance to Fusarium wilt races 0 and 2 and 0 and 1 respectively. And we have the FAT gene. It is a pleiotropic gene, what means it confers two types of resistance, the resistance to the colonization by the aphid, but also the resistance to the virus transmitted by this aphid. So in order to start a breeding program, we have to know which cases of the pathogen we have in our fields and in our growing region. This is of substantial significance, both for combating the disease, but also for the breeding purposes. And to identify the different races of Fusarium wilt and powdery mildew, this can be done identifying the level of specific resistance of melon differential cultivars that are used for each pathogen. For the case of Fusarium wilt, 4 races are described worldwide. We have 0, race 1, race 2, and race 1.2, which is the race the most virulent one. And here in Tunisia, the 4 races were identified with the predominance of race 1.2. And these differential cultivars are used in order to identify the different races. So the interaction between the gene of resistance existing in the cultivar and the race can allow us to define specifically the resistance, the race of the pathogen. For powdery mildew, I think 28 races were described worldwide, all over the world. But here in the Mediterranean region, the most extended races are race 1, 2, 3.5, and 5. And these are the different differential melon cultivars that are used in order to identify the specific race in the growing region. So in order to screen for resistance to biotic stress, we usually use artificial inoculation, making the host plant with the pathogen, and we observe symptoms. So from the beginning, when I started the program, I used to do artificial inoculation. And then when I started collaboration with the Spanish team, so we used molecular markers, which is more effective method in order to identify if the gene of resistance, the major gene I've told you before, from 2, from 1, and the gene, are in our local land races or not. In the case of powdery mildew, there is no universal molecular markers, so we only did artificial inoculation, and I will give you later more details about the different inoculation that we did. So for the results of Fusarium wilt, we have found that the resistance from one gene was present in 13 accession, and from 2 gene was less frequent and only was observed in 2 accession, 5 and 12. And regarding the race 1.2, I've said it is the most virulent one, and the resistance to the race 1.2 is different from the race 0, 1, and 2. Race 0, 1, and 2, the resistance is quantitative, but race 1.2, the resistance is, the race 1.2 is quantitative, and the area under the disease progress curve showed the great behavior of the different melon accession used, and we identified 3 accessions that are partially resistant to this race 1.2. So here, QTL mapping is interesting to be done in order to identify the genes or the genomic regions that are involved in resistance to this fusarium race. For the resistance to aphid, this insect vector of many viruses, we identified that the mature gene VAT is only present in one accession. It is this accession is very interesting. It is called Chanem. It is of ananas type of the botanical group Reticulatus. This accession has the VAT gene, but also showed the presence of the FOM1 gene when we did the molecular markers for fusarium wilt. And for the resistance to powdery mildew, so we did artificial inoculation by race 2, race 5, and race 3.5. And we found that the resistance was only found for race 2, where 5 accession showed total resistance to this race 2, whereas for race 1 and race 3.5, there was no resistance. And in order to confirm the results of the artificial inoculation, we observed the symptoms upper natural infection according to the leaf area covered by the powdery mildew, and we observed that these same accessions that were resistant by artificial inoculation were also resistant under natural infection of the disease. So from this study, we were able to conclude that race 2 of powdery mildew was the race most prevalent in our condition and the natural condition of infection. And after analyzing the various components with the various genotypic variants, the phenotypic variants, and the environmental variances, we see a very strong genetic influence on response to powdery mildew, to these race. And the most important thing that this resistance was found in accession that as of inodorous and reticulatus horticultural groups. And these two botanical groups are appreciated by the consumers because they have good quality. In general, and in literature, the resistance found in all over the world for the powdery mildew is identified in accessions from India. And we also concluded here that the resistance to powdery mildew race 2 is race specific, and we started a preliminary study of the inheritance of resistance, and we found one gene that is implicated in the accession that showed resistance to this race. But the most important thing here is that the Chanem accession, do you remember? I told you before, it has the VAT gene and FOM1 genes, but also it is resistant to powdery mildew race 2. So these interesting accession have multi-resistance, and you can ask how was this possible, all these gene of resistance in the same accession? Well, since the advances of Melon genome sequencing by the Spanish team in 2012, this has shed light on the fact that the region containing the VAT gene is very rich in gene of resistance. So this accession, Chanem, could have other gene of resistance. That's why genome sequencing is essential to uncover all the genetic information it contains this accession. So once we identify the resistant accession, and the next step will be the utilization in breeding program. So what I've showed you till now is the pre-breeding phase, because in breeding we have 3 essential phases: the pre-breeding, the breeding, and post-breeding. In the pre-breeding we have the collection and characterization, the evaluation, the screening, the genetic study, and so on, and what it was done till now. And now we're gonna start probably the breeding, which include all type of crossing, the generation advancement, if the method of selection that we will use, and the method of breeding that we're gonna use, and then the post breeding. which include on-farm trial, the variety release, and the maintenance breeding, and so on. So in our case, when we identified, we did the genetic diversity, when we've shown the great extension of the variability and the genetic diversity existing in the germplasm. We identified also several sources of resistance. We found several genes of resistance in different accession. So now to start the hybrid breeding properly, and we opt for hybrids. So why hybrids? Even it takes longer time to achieve the hybrid development, but it is preferred because mainly we can exploit the heterosis. The heterosis is the hybrid vigor, which is the greater performance of the progeny in comparison to their parents. And this can be for several traits such as yield, resistance, and so many other traits. And also the hybrid varieties ensure the uniformity of the harvest and the greater yield. So that's why we appreciate hybrid breeding, hybrid varieties. So to develop performant hybrids, we have three main phases, starting from the point of developing homozygous inbred lines. And to achieve this, we have to make self-healing for several generations, from 5 to 7 generation, then to make crossing of the productive inbred lines. Here the goal is to make the crosses of good by good in order to have the maximum of heterosis in the progenies. Then the hybrid development and evaluation that I will develop later. But let's here see this hybrid breeding and development is dependent of the hand emasculation and pollination. This manual crossing is needed to ensure the success of the different crosses, and which need obviously high technicity, and it is labor intensive. As so here are the 7 parents that we selected from the pre-breeding phase. So these parents have all gene of resistance, or good quality, or moderate or good yield. So from this point, we will make the development of inbred lines, which is the first step to start to create the homozygous inbred lines that we will need later for their hybridization. So for the hybridization, we have several types of crosses that can be done, or Diallel cross, or line per tester cross, or whatever, in order to produce F1 hybrids. But in my case, we use Diallel cross. And then when we make these crosses, we do the Diallel analysis in order to identify the best parents and the best combination, measuring the general and specific combining abilities and the heterosis. And then when selecting the best parents and the best combination, we start making the hybrid evaluation and the selection of the best hybrids through field trials, multiplication testing, and then select the best one. So here we have, after making the homozygous inbred lines, we start the Diallel cross. The Diallel cross is a method where each selected parent is crossed with every other parent. So this Diallel cross is one of the most powerful tools for its way to create a matrix of all possible hybrid combination among a group of genotypes. And here we have the 7 inbred lines that are crossed with other 7 inbred lines. Okay, and here just to mention that we have 2 types of Diallel cross. The half Diallel cross that I used here in my breeding, it is half Diallel or incomplete Diallel without reciprocals, and we have also the full Diallel that is used when the reciprocals crosses are done. But the full Diallel is used when we have maternal effects of the traits in the concerned traits. So in our case, we used half Diallel cross. We obtained at the end 21 crosses combination for the F1 hybrids, And these 7 embryo lines were produced by self-finish. They were placed and tasted, and the Diallel analysis that we conducted allowed us to define the general combining ability, which is related to the breeding value of the parent, and it is associated with additive genetic effects. We have also the specific combining ability, which is relative to the performance of a cross, and it is associated with the non-additive gene action. We have also estimated the broad sense heritabilities, the heterosis, both for mid and best parent heterosis were estimated. And here are the results of what we did. So for the combining ability values that were estimated for all the parents and for the different fruit traits trait. And from this study, we were able to identify which are the promising parents that are consistently contributing for the beneficial trait. So here, those traits or those parents that have high positive general combining ability are chosen as parent for breeding programs aimed at improving fruit weight or whatever the trait that it is need to be improved. And this general combining ability indicate a strong additive genetic effects of the traits that we study. For the specific combining ability estimates, as shown here in this table, so it was estimated for the different combination of the different crosses, and this allowed us to determine which specific hybrid combination was the most outstanding performance. So here we selected the crosses that have positive specific combining ability. These are chosen to continue the breeding program. So also the evaluation of heterosis were done, and here we identified the most performing hybrids for key traits, especially fruit weight, total soluble solids, and precocity. And here we observed that for most of the traits and in several crosses, we observed the effect of heterosis, and this is what we are looking for when we are creating or we are developing hybrid varieties. Taking correlation into consideration is also important for every breeding program, because we have to know which are the correlation and take into account the indirect selection in case of we have two traits that are not correlated, not positively correlated, and we need to take that into account. So this here is a wrap-up of the hybrid breeding pipeline. So we start from developing the Melon embryo lines. We control the crossing experiments, making Diallel or line cross tester to generate Watman hybrids, to evaluate the combining ability and heterosis, as I told you before, to start to analyze hybrid performance. But before this, we have to select the best parents and the best hybrids. Where we are in this step. So we are selecting the best parents and the best hybrids in order to test their stability and performance in multi-location, and they will be grown by farmers in different locations. We can also introduce more parents in this pool in order to make heterotic groups and enlarge more the initial land races. So here, once we identify the best parent and the best hybrids, and we select the best hybrids that is stable, highly performant, and we confirm what we were looking for. So we start with the variety registration in our national variety catalog. But before this, we have to make the DOS test. This means the distinction, uniformity, and stability test. If we succeed in the DOS test, so then we gain the official approval for commercial release. So the work is ongoing, and the expected impact when we develop at NBN, we create these hybrid variety, it will be the development of high-performing local hybrid melon varieties. we will gain and have an improved yield quality and disease resistance. We also going to supporting the regional agriculture by providing high quality locally bred varieties. But above all this is to save hard currency paid for importing seeds, because most of the time we use import seeds for several crops, not only in melon. So developing our local hybrid or our local melon varieties with good characteristics will benefit the government, the farmers, and all the stakeholders. So here's just an overview of also what we are doing regarding the microbiome diversity in order to include this in our breeding program. Is another aspect that we are studying in my research center, where my colleague of soil science, soil science researcher, is studying the microbiome diversity. And in our cases, we wanted to know the microbiomes associated with melon landraces and with melon hybrids in order to be taken into account in future breeding programs, because it is now an emergent field. So the first preliminary studies showed that the great diversity, obviously, of the different microbiomes and microbes associated with the different melon genotypes. And the results here show that the great test of average fruit weight and yield was associated with the highest actinomycet bacterial, Trichoderma, and Aspergillus. And we will develop more this aspect in order to include it in further studies. So what are the concluding remarks? So we have showed here that our melon germplasm holds immense potential for melon breeding. We have shown the great diversity and we have shown that several land races have gene of resistance for the major abiotic stresses. It will be interesting in the future if we evaluate also this germplasm for drought and water scarcity, because nowadays with the threatness of the climate change, and in Tunisia we are among the most countries threatened by this effect. It will be interesting if we valorize this collection also for the main major problems here in Tunisia, which is drought and water scarcity and also salinity. So we have shown also among the challenges in conventional hybrid breeding, as I've shown you, is the high breeding cost and long development time. The pollination control is crucial to maintain purity. Hence, emasculation and pollination are labor intensive and costly. So in order to overcome these challenges, modern approach to accelerate hybrid breeding is crucial. So we have, for example, the double haploid, which is the pollen or ovule culture in vitro, and this allow also the genomic selection, and this allow to shorten the breeding cycles and to gain in time. Male sterility is very interesting, and it should be applied because it is an efficient alternative for hybrid seed production, and it helps a lot once making the hybrid production. So strengthening regional and international partnership is crucial to advance melon breeding. So it is really needed also. Just to mention that this research is collaboration with the different institutions at the national or international level. I want to acknowledge all the researchers from different institutions, from my institution, from the national other research center or other institution in Spain or Germany or Egypt. With all their collaboration, we were able to achieve these results and these good results. Also, the person behind the scene, I will not forget the technician of our experimental station that are taking care of our experiments. Also, the different students I have supervised during this research. And thank you for your attention, and I look forward to your feedback and if you have any question. Thank you.
Thank you, Hela. Thank you so much for a wonderful presentation. We have several questions already in the chat. If you want to stop sharing so we can focus on the questions. I'm gonna use my coordinated role to ask the first question. Are these land races still existing in farmers' fields, or is just part of collections? Is everything else on hybrid production? What is the status? I mean,
the start of the Germplasm collection, you mean, no?
yeah, the
Right?
status of these land races. Are some of them still held by
Yeah.
farmers, or is just part of a collection?
Yes, when I started making this breeding program, we started from contacting local farmers in order to collect and take from them the seeds or the fruits to be evaluated later on. And we started from the local farmers and we collected what they have in their fields. And we started evaluating them in order to be sure of the characteristic of the botanical group and so on, and to evaluate them and to valorize them as I've showed you.
Okay. Yeah, I'm amazed by the diversity you showed. Here in ours, I mean, we're lucky to find 2 or 3 varieties of melons when we go to buy. It's amazing what we are missing in terms of the diversity of types, and amazing. Okay, let's go to questions. Pranjana is asking what other methods can be used for artificial pollination other than hand pollination?
Rather than hand pollination, as I said, the male sterility can be used to produce hybrids. But in case of melon, we are using the cross manual crossing and making the cross. It's labor intensive too, but we can, as I've said, using male sterility, it will help a lot the development of the hybrids.
How about the manipulation of the sexes with hormones? I understand that in larger seed production fields, they use either gibberellic acid or other hormones to convert plants to full male or full female.
Yeah, I heard about this, but in our cases, we use conventional methods intending to make the things as safe as possible. So we do what I've showed you, just making process at the small level. And later on, if we will enlarge for the production and so on, I'm thinking about the male sterility, which is it's genetically inherited, or as we all know, the male sterility is a mitochondrial genomic effect. So we can look for a potential male sterile line of melon that can be used in order to avoid making emasculation and so on.
Sounds good. Also, Pranjana is asking, what tools do you use to determine general and specific combining ability?
We use Griffin model. We can use it with different or R statistics and using statistical analysis. Okay, and for the Diallel analysis, we have it depends if you have self-link or no, if it is full or incomplete Diallel. Depending on that, and we have the briefing methods, and from the briefing methods, before this was it was doing by calculating and by hand, but nowadays with the progress of the statistical analysis, this can be done by the statistical analysis, and she can learn our paper. The last one I've showed on combining abilities in Melon, where we developed very well the methods that we use for evaluating and estimating the general and specific combining abilities. And we used the ARC program and the different program architecture.
Thank you. Prince is asking, I imagine you do include checks, commercial checks in your evaluation, F1 evaluation?
Commercial checks, of course. We have to put an hybrid commercial one in order to see where we are, because we have to have a control one to see where we are okay or no. But the most important thing, I mean, we cannot compete with the higher companies that are producing hybrids, you know, because they are using more technologies and more sophisticated technologies, and they can get the gene in question, making, for example, gene editing, or I don't know. But in our case, making conventional methods, if we succeed to obtain an hybrid variety, even it is less than no more productive than the hybrid the commercial one, even less. But if it has resistance and if it has good quality, so we have a balance there. I think. But
okay.
An economic study should be done, but I think there is a balance there that can be achieved. But we cannot obviously compete with the big companies.
I'm gonna jump to the end of the line in terms of questions to give a fair share to everybody. So Pablo Rios is asking if your collection of local germplasm included wild, non-cultivated melons.
No, I didn't found. When I started collecting, I only found these the botanical groups I've mentioned. But for other crops, for example, for watermelon, I found relative species and wild species. The bitter melon, the colocanthus for watermelon, I found colocanthus. And I found also another for this cucumis melon, I didn't found, no.
Okay. This breeder from Ethiopia is very interested in connecting with you. Solomon is asking if he can have your email address. Is it okay if I post it in the chat, Hela?
Of course.
Okay.
This is
right, Solomon, there is Hela's email address. Okay, please reach out to her.
I'm
go ahead.
Open to any collaboration, and I'm open. Of course, this is the target of presenting our research in order to connect with other people, and if we can make collaboration, it will be great.
Very short but very interesting question by Stuart Garner. Do you evaluate the taste of the melon? So how is that part of the organoleptic and preference traits done?
Yes, very good question. For the organoleptic, I only evaluate the most appreciated things by the consumers, which are the total soluble solids. So measuring the degree Brix, which measure the sweetness of the fruit, and also the firmness. The firmness, because it's very important, because if crunchy fruit, it's for me, for example, I prefer crunchy fruit, and it depends. For the firmness, it depends on the preference of the consumers, okay? But these both traits are evaluated, and later on maybe we can evaluate which are the polyphenols, which are for the local and races, we evaluate the antioxidants and the vitamins and so on. But for the hybrids that we already obtained, we didn't yet did the vitamins and polyphenols. But for the total soluble solids, it's okay.
So do you have a panel evaluation by panel, a panel of consumers, or that give you a sort of a rating of your materials?
Yes, I will do it. Yes,
you will do it.
Not yet, but yes, it is in my program to do that, because we need to know the appreciation of the consumers, because at the end, this product is going to the consumer, and if not appreciated by the consumers, it will not be commercialized, no? Because for the farmers, he's looking for the yield, for for the resistance, in order to not use chemicals and so on. But for the consumers, we have to take into account also their preferences. And the preferences, it depends of, but the most important, there is a criterion of TSS that we don't have to be under the 12-degree Brix, for example. So we have to be sure that the sweetness is there, okay? And other preference, for example, crunchy, not crunchy, other details, it depends of the appreciation of the consumers. But yes, a panel will be done in order to evaluate all the different characteristics, organoleptic characteristics.
Okay. A question is about how easy it is to exchange germplasm with Tunisian institutions. I mean, if you want to exchange with another country, it's pretty relatively easy. I mean, as a protocol to follow.
Not easy. Things are changing here, so maybe because a few years ago it was so complicated, we cannot take away the even in research, okay? But nowadays things are changing, so we have a mutual transfer agreement, I think, something like that, and we have to see with the ministry how are the things going on. But for example, if I want to import from other countries, for example, I collaborated with the Spanish, and they sent me, for example, some materials, especially the differential cultivars that we use to identify the different races of fusarium and the powdery mildew. They sent me the different differential cultivars of melon, and they used them. And this was done by the MTA, I think, mutual transfer agreement. But on the other side, from Tunisia to outside, is more complicated. It can be done, but it's very complicated, and a lot of papers and a lot of administrative things.
I see. Okay. David is asking if you can develop a bit more on the FON 1.2 gene. Is it a dominant gene, or how many QTLs are known? He understands that this is a quantitative trait. And have you bred with it? How challenging would it be to breed with that gene?
So he is asking for FOM1, the gene.
1.2, yeah, 1.2.
So let's for fusarium wilt we have 4 races: race 0, race 1, race 2, and race 1.2. Race 0, 1, and 2, the resistance to these races are qualitative. We have 2 major genes I've mentioned, for F11, which confers resistance to races 0 and 2, and F12, which confers resistance to races 0 and 1, okay? For the race 1.2, it's the most predominant one and the most virulent. And it is quantitative. All over the world, studies have been done regarding the QTLs mapping that have done. But in my study, I only did artificial inoculation. And I'm planning to do cultural mappings in order to reveal the more details about the accessions. Only for accession, they showed partial resistance. So I need to do the cultural mappings in order to reveal the genomic region and how many cultures are involved there. So I was stopped there. That's why I put it in my perspective to be done.
I suggest David to connect with you to continue exchanging on this topic. Okay. I think there's one more question here. It's about the half diallel at the possibility of missing some of the senior or maternal effects when you use half diallel instead of full diallel. Do you have any comments? Because he's saying how significant is that with that loss?
Yeah, if I understand well the question, so he's asking if I use half Diallel, I must miss some maternal effect. No. So that's why when the trait that we are studying, we need to know about it, if it is maternally inherited. In the case of melon, I know that the male sterility is the only trait to the best of my knowledge, is the trait that is maternally inherited. In the case of fruit traits like weight and so on, in general and in the literature, we usually use half Diallel, and even when they use the full Diallel, they found that there is no maternal effect, because when we did, if you don't know the trait in question, you are doubting if this trait has maternal effect, so you can do it, the full Diallel, you can do it, and you can making later the analysis, you will detect if it has maternal or no maternal effect. But depending on the literature done, the traits that have evaluated for the half Diallel are not don't have maternal effect. So that's why I did half Diallel.
Okay. And also one of the last questions is how you manage the difference in ripening time, on flowering and ripening time when you decide what parental lines to cross.
Yes, very good. Very good, very good. This depends of the botanical variety, okay? Because some varieties like reticulatus, I've showed you, when it has smelling, so when it starts ripening, we have a smell, so I know. And also the peduncle, when the fruit, it is detached, so it is detached. But some other botanical groups like Inodorus, we only see the ripening, it change of color. It was green, so it moved to the yellow, so we know it is ripened yet. And sometimes we can do TSS in order to see if it is okay or no. But visually, is we can do it visually, as I've said you, depending of the botanical variety. Some botanical varieties like Inodorus, these change of color, so we know that it will be because the peduncle is not detached from the fruit when it is ripened, okay? And we have cantaloupes or reticulatus when ripened, so the peduncle is detached, okay? And it smells also.
Okay, just a recommendation here from Hamdino to try to cooperate with Uzbekistan. They seem to have amazing diversity too. Okay.
Yeah, Pakistan, India, they have great diversity.
Yeah, all Central and
melon.
Central Asian countries. Yes. All
Yeah,
right.
yeah. Thank you.
Yeah. Okay. So thank you so much, Hela, again.
Thank you.
I appreciate
Thank
your.
you to all of you. Thank you very much.
Let me. Thank you for everybody that attended this seminar, and we look forward to have you with us next Thursday. We have our guest is going to be Dr. Peter Cousins from Gallo Wines here in the US, talking about improving grape and wine quality through plant breeding and genetics. Thank you so much, everybody. I hope you guys have a great weekend. And tomorrow, with the announcement of next week seminar, there will be a link to today's recording already. Okay, so thank you again, and I'll look forward to see you again next week. Okay, thank you. Bye.
Thank you. Bye.